Anyway, so I'm going to tell you a little bit about some work I've been doing with a bunch of people, and here are some of the names. So Che Yu was a PhD student at UC Irvine. He was co-supervised by Hernando Bao, who's also at Irvine, and myself. She's now at Apple. Cheng Han is a current PhD student at UC Santa Cruz, and then Dan Rowe, they are involved in the second part of the project that corresponds to the second part of my talk, and I hope to get there. So I'm just going to give you a little bit of background on fMRI data, and then we'll talk about some models that are region-specific. So we'll talk about that first, and then we will talk about some other kinds of data where the fMRI images are actually complex valued, and this is a little bit more recent. So just to begin with the kind of data we work with, I don't know how familiar you are with fMRI data, but let's assume that we are starting from the beginning. So the idea here is that they are based on the so-called blood oxygen level dependent response, and when you have neurons that are activated, the requirement, the metabolic requirement for oxygen increases, and you're going to be able to see that in terms of changes in the magnetic resonance signals. So these are, you can think of them as images over time that are collected during a particular experiment, and they have very good spatial resolution and relatively okay temporal resolution. They are not so great in terms of the temporal resolution. So the kinds of questions that people are interested in addressing here are related to activation, so which areas of the brain or which voxels are active during a particular experiment or a particular task, and also what is the connectivity between, it could be between different brain regions or between different voxels if you're working with a whole image. So those are the type of questions. So you may have seen pictures that look like these that indicate particular regions that are active in the brain, or just in terms of connectivity, which areas are connected. So the way these things are modeled, you usually have an experiment, so you can think this is a little toy example. You have here a particular stimulus that is given to the subject. The stimulus is on and off, right? And then what they do to model the ball response is to think of it as a convolution of that stimulus signal with a so-called hemodynamic function. And what you get is essentially something that looks like this, and then you're going to be comparing the amplitude. You can run some kind of regression model on this and what you observe. And then you're just looking at what is the amplitude that you get for the particular region or voxel or whatever it is that you are analyzing. So in terms of connectivity, people are interested in two types of connectivity. One is the so-called functional connectivity. You can think of this in terms of this picture. If you are collecting the images over time and you have a particular region in the brain, I'm calling this one P and this one Q, then they are related. Whatever happens in one of the regions is related to whatever happens in the other one. And then there is effective connectivity, which is the directed influence from one region to another one. So you think of the temporal connection. If this is a particular region in the brain at time t-1, you're going to think about what is the influence of that region at time t. So those are the two types of connectivity. Now, in terms of activation, and we will see the two types of analysis in my talk, when you work at the voxel level, it's a very big problem because you have lots of voxels in the image, but you can think of this as a regression problem at the voxel level. So the idea is you have your observed image here at voxel V and time t, and then you can run this regression where this could be some kind of trend component that you have in the data, and then this is the expected volt response for a given hemodynamic response function, okay? And then you're going to determine if the voxel V is activated when this coefficient is different from 0, okay? That's a way to think about activation. So this is a little toy example. If you have ever played Minecraft, my son plays that. This game with pixelated images. He always says, "Oh, Mom, that looks like the brain of a Minecraft guys, so they are just pixelated. So here is the little toy. So this is just the experimental design. The stimulus is on and off. I combine that with the canonical hemodynamic response function that I'm not showing. You get the bold. And this is the activation, the true activation image here, right? So these are the areas that are active, and the strength of the activation is different for the different voxels. And when I look at the time series at a particular voxel, in an area that is not active, say here, I observe this, and if the voxel is active, I should observe something that looks like this. And what I'm recording are these images over time, so there is noise added to that. And the noise is complex. Here is, the structure is very simple. It's just to show you a little bit how this works. So the idea here, there are many challenges and many questions to answer, as I said before. So you have, the number of voxels is very large. These are huge images, and when you're trying to, say, detect activation, you have to think about issues like multiple testing and all these other things. They have a very complex spatiotemporal structure. The temporal, you could think of, well, the image at time t is related to what happened at time t minus 1, okay? And also, temporally, you are thinking, well, the brain regions are related, right? So there are lots of different, very complex models. The computational complexity is an issue, because if you come up with a very sophisticated model that you cannot really fit, then that's a problem, okay? So that's another thing to keep in mind. So when I was telling you that people use here, you're convolving the hemodynamic response function with the stimulus, well, what hemodynamic function are you going to use? That's another question. So many analyses just assume there is a single hemodynamic function. It's the same for all the regions. But in reality, you know, first of all, there is uncertainty about those things. Second, it's not reasonable to assume that you have the same hemodynamic function for all the brain regions or the voxels and all the subjects. The second part of my talk, as I said, we're working now with fMRI data that are actually complex valued, meaning that you're going to have a magnitude signal and a phase signal, or a real and imaginary part, however you want to work with it. Most of the fMRI studies use the magnitude data only. So what happens is, if you're a neuroscientist, you're using your machine, what you get there is just the magnitude. They don't even get to see the other piece of the data, so that gets discarded. So we are interested in just analyzing this data. And then the question is, well, what about connectivity? All of this I was talking about is in terms of the activation. So this is some work we did at the region-specific level. So this involves a multi-subject study. We have several subjects. Part of the group was a group of patients who had a stroke 6 months prior to the study, and then they were asked to do something with the part of the body that was affected by the stroke. And then we also have some healthy subjects. So it involves multiple subjects and groups, and this is joint work with some people at UC Irvine. And the connectivity, in terms of the elements of the model, we define it through a vector autoregressive component with spike and slap type of priors. Then we have group-specific activation and connectivity parameters. One of the important features here is this thing. We are assuming that we don't know what the hemodynamic response functions are, and we're going to assume that they are subject-specific and also region-specific. We did extensive simulation studies just to see how much this affects the uncertainty, say, about the HRFs affects your inference in terms of the activation and the connectivity as well. And we also looked at the stroke data. So just to be a little bit more specific about the data, we have 29 patients who had a stroke 6 months prior to the study, and they have residual motor deficit. So we have people that got this right side of the body affected, meaning the stroke was on the left side, affected the left side of the brain, and then we have 13 people on the left side. And we also have 13 healthy subjects. Because of the way we have the distribution of patients, we had 15 of the right side affected patients were also right-handed. And we have, in the healthy group, we have 12 that were right-handed and only 1 that was left-handed. So we're going to use only the right-handed patients in this particular case. As I said before, if you have a stroke on the left brain hemisphere, that's going to affect the motor deficit on the right side of the body. And what the subjects were asked to do was to perform this task that was consisting of just having the hand of the affected side of the body do the grasp and release movements. And in the control, you do the rest. So the experiment looked like this. They had 3 sessions. They ran 3 sessions for the stroke study, for the stroke subjects, sorry, with 48 fMRI runs, and they had 12 runs alternating within conditions. So they started with the rest, then they did 12 with the task, and then 12 resting, and so on. And this results essentially in 144 scans per region of interest. As I said before, in this data, we are not working with a full image. We are working with specific regions that were selected by the neuroscientists. And I'll talk a little bit about that. The time resolution here is 2 seconds. That means you're going to have an image like this every 2 seconds. And these are the pre-selected brain regions that the neuroscientists took. So there are the so-called primary motor regions. There is one on the left and one on the right side of the brain. And these are the regions that are supposed to be the ones responsible for the movement of the right hand and the left hand. So if you're a healthy person and your brain is not affected, you should see these areas be the areas that are mostly involved in the task. Then there are secondary motor regions. There is one in the left and one in the right, and then there is one called the supplementary motion area that's in the center, and this is responsible for more advanced functions. So as I said before here, if you are a healthy subject and you are working with your right hand, what you expect is that the LM1, the region on the left, the primary one, is the one that is involved. So this is a little example. So this is a picture of the regions that we are considering. For each region of interest, what they did is they took the average across the voxels, the average fMRI across the voxels in those regions, and then this is how the time series looks for, again, a healthy subject performing a task with the right hand. This is the LM1, so you see here, this is when the stimulus is on, off, on and off, and this is what happens with the RM1. So this looks a little bit like what we were describing before. So the goal is, how does a stroke affect the brain in terms of the motor function? Do the healthy patients look the same as the stroke individuals? So what we did here, this is a model, so we're assuming we have 2 conditions. You could have an experiment with multiple conditions, and they have experiments like that when the subjects are performing multiple tasks over time. And we have a certain number of regions. We ran this with 3 regions, 5 regions, and a few more regions. There is the signal is subject specific, and then we have also different trials per subject. So what we have here is your typical kind of regression model, right? When you look at the activation, is the coefficients here are subject and region specific, and then this is what comes from the excess here is just a result of convolving the hemodynamic response function with the stimulus. But we don't know the hemodynamic response function. So what we are going to do is we're going to try to represent this, and we have done different kinds of analysis. This is the one we settled on, essentially just representing these as basis functions. Here, the matrix here, this H is just some basis vectors, and there are some that people use a lot in modeling these kind of signals. So again, what you're doing is there is uncertainty. This is something that you want to discover for each region and each subject. You convolve that with the stimulus indicator, and then you put that into your regression model. In terms of the basis functions, you can look at the details in the paper, but essentially they are rich enough that with a small number of bases, you're going to get most of the variability observed in the data. This is just the first few bases, and these are just some HRFs generated from the basis function. And in terms of the connectivity, as I said before in the elements of the model, we model this through a vector autoregressive model. And because these are fMRIs, usually the order of the vector autoregression is very low. You don't need to go back in time too long. So we have some vector autoregressive models, and for us, the parameters that are going to tell us about the effective connectivity are the vector autoregressive coefficients there. And so we have a spike and slab structure. And then for the functional connectivity, we are looking at partial correlation inferred from the model as well. So how do we build the priors? Well, we have information from the neuroscientists about things like these parameters that I'm listing here. We have information about the time to peak, the time to post-stimulus, the height of the peaks, what is expected here. And what we do is we feed the prior distributions, we simulate from those prior distributions and see if they are reasonable, if they are giving us shapes that are not too crazy in terms of where these peaks lie and where this peak goes. So we cannot get anything here, would be very unlikely. Okay, so that's my prior distribution. And for the remaining parameters, what we are doing is we're using just your favorite conjugate prior distributions, and then we just do posterior inference with all these quantities. Oops. So just get back to where we were. So what kind of quantities do we infer here? So we infer subject and region specific parameters such as the activation strength, the connectivity strength, and also, very importantly, the hemodynamic response functions. We get posterior distributions on those. We get group-specific inference in terms of the mean activation strength, group connectivity, and mean connectivity strength, and we also get the mean for the group, the HRF function for the group, and it's also specific to the region. What do we do? I'm not going to talk about posterior inference. Here is your standard MCMC customized here, and then there is also a little bit, we did some variational base just to look at approximations in case you are considering very large number of regions. We looked at several simulation studies. This one involves 30 data sets in 5 regions of interest in each of the data sets. This is a true network that you have in terms of connectivity, and 2 of them were not activated, the rest were assumed to be activated here, and these are the HRFs. So I have 5 of them, 2 of them have essentially the same HRF. There is one here that probably you cannot see, and some of them are similar and some of them are very different in terms of where these peaks are. So there is uncertainty again. We are trying to infer those things. So we looked at just the results in terms of activation and connectivity. We got very good results. This is posterior inference on those HRFs for 3 of the regions. I'm showing you here region of interest 1, 2, and 5, and there is more uncertainty for some of these because you have less information in the data about this distribution, about these hemodynamic response functions. One thing that is important that we learned from the simulation studies is how much as I said, in most of the models, people just assume I'm just going to use the canonical hemodynamic response function and run my model and see what happens with my activation and my connectivity. What happens is the impact in the results in terms of the activation and connectivity is huge. Even if you're considering hemodynamic functions that are not too far away from the truth, you can have very big differences in terms of the inference that you get here. So that's essentially, this is again comparing MCMC with variational base. I'm not going to talk about this computational part. We looked at some data. So this is, I told you that for the multi-subject analysis, we were only going to work with the right-handed patients. This is the analysis for a left-handed patient that had motor deficit on the left side of the body. And these are the hemodynamic functions that were inferred from the model. This is only involving 3 regions of interest, and you can see there is a big difference. So most analyses, again, assume my hemodynamic function is the same for the different regions in the brain. And this is what you infer from the model. They look fairly different for the different regions. Now, the multi-subject analysis, we ran with the 15 patients who had a stroke and were right-handed, and 12 patients that were healthy. In terms of the connectivity, this is what we discovered. So these are my connectivity graphs based on the posterior inference. And what we can see in general is that the connectivity within the primary motor region appears in both cases, but we see a lot more between ROI connectivity in the healthy subjects than what we see in the stroke patients. So this is just, again, inferred from the model. In terms of the activation strength, we see that LM1, again, is the most active region of interest for both the stroke and the healthy patients, but there is an increased activation here in pretty much all the regions, and in particular, there is a lot. This one is a very highly active region that is not so active in this particular case. So we can also compare the different estimated hemodynamic functions. I'm showing you here the posterior means for the 5 regions and the healthy versus the stroke patients. And what we see is that there is a lot of variability here, so it's important to have subject-specific HRFs, but we couldn't find anything that we were able to distinguish in terms of, well, the healthy are very different from the stroke patients in this particular case. There is a little bit more variability in the stroke group, but that's essentially, in terms of the hemodynamic functions, this is what we could find. So in terms of what the features of the model are, we do simultaneous inference of HRFs, activation and connectivity. We have condition-specific effective connectivity parameters, and we also have some findings in terms of the regions that we think are most are kind of activated in the patients who had the stroke to try and compensate for the damage they have in the brain. So all of this is in the paper if you're interested in looking at this. So the second part of the talk, so this was again using data region specific. The second part of the talk is let's look at voxel level data. So I'm going to think now about the entire image. And as I was saying before, pretty much all the studies you read about are dealing with magnitude-only data. So you're using this image, and then you're looking at activation and connectivity using those data. So what's happening is this piece of the data gets discarded. This is the face. You can also think of it as, well, this is my real part of the image and my imaginary part of the image. So most studies just look at this. The machines, as I said before, don't even give you this as an output. So we are looking at some data where we have essentially these two. And Dan Rowe, who is our collaborator, has been advocating for the use of these complex valued models for a long period of time. And we are looking at this type of data and just how to look at activation from these models. So he considered, and he and his collaborators considered models that look like this. So you have your complex value image, and then you have a phase essentially parameter, and then you have a magnitude parameter, sorry, and a phase parameter. And then you have your activation you're going to be modeling in this fashion, right? So we start in the data that I'm going to show you right now. We are going to assume that the phase is constant over time. So we only have this parameter here. All of this disappears. And then I have my magnitude parameters in here. So you can write this thing as a regression model at the voxel level. So I have my entire time series for voxel. This is my fMRI time series at voxel V. And then I have a design matrix that has whatever you need to have in there, all the HRFs, the all the ball signals, all the trends, whatever you need, and then you have this complex valued term here for the noise. So, as I said before, if you look at the literature, you have very sophisticated spatiotemporal models that are available only for magnitude data. And these models that were developed by Dan and his collaborators are models that use maximum likelihood estimators and all kinds of things, including likelihood ratio tests. What they do in practice is they correct for multiple testing using either Bonferroni or FDR. And what we are trying to do here is we looked at some of the variable selection methods and just essentially use them in the complex value domain. So this is what we are doing here just in terms of detecting activation. So this is how our models look like, right? So I have my time series here with the signal observed at voxel V. And you can, here I'm assuming that the complex normal distribution is the simplest possible, so you have circularity and independence and all you need to have here, but you can consider more sophisticated structures, and we just, in order to decide if a particular voxel is active or not, I'm going to use both pieces of the data, the real and the imaginary parts. So this is how my distribution is going to look like. So it's the usual type of distribution, but now we are using the complex valued signals. And then we have just the standard construction here. I'm going to skip over the algorithm, and I'm going to show you some pictures and some results in terms of simulation studies. So here is the simulation study. Again, this is in equations, what I showed you in a picture before. So let's just stay with the picture. I'm going to take this data, essentially. This is the true, and I'm going to run this with the models that we have, right? So the data were simulated using a package that is called NeuroSim that allows you to just even add noise and additional terms that are more realistic in neuroscience studies. And these are, we consider different kinds of models. I just want to show you some of the results, and I'm not sure how much you can see from this. But essentially you have, there are some voxels here. This is the models. I have the complex models, two types of complex models in the upper row, and then these are the results for the magnitude-only data. And the magnitude-only data, the model using the magnitude-only data, they do worse than the complex-valued models. And these are models, we ran these for different signal-to-noise ratios, and this is for an intermediate kind of signal-to-noise ratio. Then we said, okay, this is great, but let's try this on more realistic simulated data, and also let's try it in real data. And this is, I'm going to show you those two cases. So Dan has a way to simulate realistic, more realistic data using some physical parameters. So what they do is they use a discrete version of the magnetic resonance signals equation, and they have some input parameters here to model some characteristics in the brain, like, for instance, there are parameters related to what is going to be gray matter, what is going to be white matter, and some additional parameters. So these are, all of these three parameters here are the ones that go in the equation. And then they add noise to that, they generate those signals, and just in pictures, these are essentially they combine essentially these images that come from the physical model, and you obtain these as your magnitude and phase baseline. These are the activated regions, so we're assuming that there are these two very small regions are the ones that are going to be active. Everything else is not active. This is the complex, and this is the, this is the real, and this is the complex, or the magnitude and the phase. You combine these things, you add noise, and this is the data we got. Okay? So we didn't simulate data from the model. This is just model we got from this physical parameter. So when you run these things, if you just look at threshold values, so again, this is a picture where probably you cannot see, there are a bunch of red and blue dots that you probably cannot see there. There is a yellow piece here, and there are a lot of false positive here, and also inside, in the region inside the brain. So even if you correct for multiple testing here using different techniques, you're going to get a lot of false positives, or you just don't detect anything depending on the technique that you use. So we tried a few different models. We tried all of them using the EM variable selection algorithm. We compared also with the magnitude-only model. So this is my first picture here, and I'll indicate there is no way to see it, but there is a few red dots here, but the model misses many of the active pieces. Then we tried the complex valued models with different parameters, and this is essentially looking like the regions that were active in the brain. Okay, so this again is to show that you miss a lot if you use only the magnitude data versus the two complex valued models. Then we ran this in real fMRI data that belongs to Dan as well. So in this experiment, what they did is they took a single subject, and it was a fairly simple finger tapping experiment. Okay, it had a block design with an initial 30 seconds of rest, followed by epochs of 15 seconds on. The stimulus was on, so the subject was performing the finger tapping, and 15 seconds off. The data dimension, we are looking at slices. You can get the 3D signals as well, and we'll consider those eventually, but this is just the slices, and this is the time component, so the signals are 510. So the time series, your time series at each voxel is this length. We excluded, just for consistency with the paper, the first 20 observations from the analysis. And again, you probably cannot see too many of the colors here, so I'll try to highlight what's happening. So this is results from their paper. In their paper, they use a complex valued model, and they did some correction for multiple testing here. Then they used the magnitude-only model, and this is a picture for the first type of model, magnitude-only model. And then they use a separate model that is taking into account the physics of the problem. Okay, so it's a physical model that is way more sophisticated and much harder to fit. Okay, and you cannot see what's happening here, so you have to trust me on this one. You can come and see the picture later, but there are a lot of false positives here that are outside the brain, so you know those are not active, and a few that appear. But it highlights this area in the brain that is expected to be active, and also something in here. But you have a bunch of false positive voxels there. Then if you run the second model, the magnitude only, you get rid of many of the false positives, but you also get rid of the areas that you are expecting to be active. And this is a physical model, and in the paper essentially they say, well, you really need to use this physical model to be able to capture what's happening. So this is what is expected essentially in terms of the activation. Now, fitting these type of models when you're dealing with these images and multiple subjects is going to be very hard in terms of the computational complexity here and how much you have to wait for the results. So what we did is we took this data and we ran the complex valued EM variable selection here for the we have again, we're comparing, these are the models, it's essentially the same pictures that I was showing you before, and these are the pictures that we get from our analysis. So this is just for comparison. So again, here there is a bunch of, just have red colors here. There are a bunch of false positives here that you don't see, so trust me on that one. Here, you don't have that many false positives. Again, you only have, but you miss many of the possibly active voxels. And then this is the one that comes from the physical model. These are our results. So this is a complex valued model. So this is actually, it should be the reverse. The first one is the magnitude only, then I have a complex valued model, and then I have another complex valued model. The parameters here are chosen by optimizing as given in the paper, just optimizing the parameters, the V0s, and assuming that the V1 is 1. So this is, again, what I want to emphasize here is that by looking at this model that does not incorporate any physics of the model and does not incorporate also any spatial structure, we are essentially discovering the same type of things that you are discovering with the more sophisticated physical model. And it's important, again, to show that just use both pieces of the data. So as a summary, we discovered that essentially these type of models are leading to an increase in power, especially when the signal-to-noise ratio is small. We have done a lot of simulations, again, for cases just for very large, very small signal-to-noise ratio, so a lot of noise. And we are working now, we are doing several things right now. Cheng Han is working on looking at a bunch of different kinds of priors and comparisons to other types of methods that are used in practice. We are also adding some spatial temporal structure to the model. But again, we have to be careful here, because if you add something that is too sophisticated from the spatial viewpoint, then you're going to have trouble essentially just fitting these kind of models. And the idea here is we want to be able to look at a lot of images from a large number of patients. So the computational part is important, and I'm actually done earlier than expected.
Raquel, I would like to know what do you think about the future of statistics in this area?
Okay, so there is a lot to be done. There is a lot of things that we can contribute to. I can tell you it's been, I've been working with different groups of people. There is a huge, there is a huge involvement of computer scientists in the in these kind of problems. I think we can contribute in lots of different ways. We are a little bit better, I think, at least for now, at communicating with the experts and at putting biological information into the models. That is very important. That is very important in this area, right? But it's a very hot area. I've been sitting in panels for NIH just working with these kind of models, and it's very competitive. There is a lot of stuff to be done for statisticians, so I always tell my students we have more data and more problems than students. So if anybody's interested in working in this area, it's a great area to get into. You have to not be afraid of the computational aspects, right? You have to just go into those things, and I don't know what your experience is. You've been working with these too. Are you optimistic or not? Who's working on this? Yeah, so there are lots of problems, but we have to. One of the issues when we talk to the neuroscientists, they are always like, "Oh, your models are beautiful. Yes, it's great, but we need something that works fast." And this is the challenge.
I think this is more related to the first part of the talk. I'm wondering whether how to disentangle the functional connectivities with the effective connectivities. In particular, I wonder whether some of the functional connectivities, they are actually effective ones, but due to the low signal-to-noise ratio, you cannot identify them as effective connectivities. I know that depends a lot on the data resolution, but I wonder if it's easy to detect that.
Yeah, okay, so that you don't have to run an exercise. Yeah, sorry about that. Just to answer your question, yes, so the idea in this, in the analysis is that we're kind of pulling information across the patient, so there's a lot of assumptions that we actually have to test carefully, because there is a lot of subject variability, but the idea here is that you're assuming that there is some information about the connectivity within the group of stroke patients, and there is also some other information that you can get from the group of healthy patients. So in that sense, I don't know if you were if your question was related to that, but yes, it's... Yes, they are learning about that. Yes, correct. But there is some information that gets borrowed from the different subjects, right? So my point is, if you are doing this for a single subject, you have to be careful, because then there is so much you can get out of the data for a single subject.
So I'll take the last question, I guess, unless someone thinks of something else. You mentioned that your constraints in the voxel problem were computational. What have you done in the way of parallelization? Yes.
So again, as long as you have a structure that is simple and you can parallelize things, this is very fast. You can do this fairly easy, right? Because everything is Gaussian, there is independence, there is no spatial structure, you can run things quickly. What you can do if you want to incorporate some additional spatial information, and this has been done by some people, is you can kind of have a model where you specify that at different levels of the resolution, and you can go with the resolution, you can choose the level of the resolution you're going to use for the spatial model in terms of your computational capacity. So this is the next piece that we are going to be dealing with. So you can say, well, you know, I'm going to work at this level of resolution for the spatial model, and yeah, if you can afford it with the parallelization of that, then it's fine. If not, then yeah, you have to go with something that is simpler.
All right, let's thank both speakers again.